It's time for us to move on to our trainee lightning talks. We're going to have each trainee talk for 3 minutes on their latest research, and then we'll have a couple of minutes for questions. And I'm going to keep time just to make sure that we stay on track because we have 11 to get through in the next hour. So I will let you know when you have 30 seconds left on your presentation. But I believe we have Miles going first. So Miles, whenever you're ready.
All right. So my name is Miles Northerly. I'm in Martha Gillette's lab, and the name of my talk is Redox in Neuronal Axons and Dendrites. So if it wants to work. There we go. Okay. So redox has a dual nature when we're talking about it within biology. So we typically think about oxidative species as being the source of evil, or sort of this Pandora's box that causes all these different problems, to include aging just in general as well. However, within biology, within the cell, it has many kind of normal typical functions, and within the brain, it's also important for plasticity and cytoskeleton, among other things. And specifically, I'll be focusing on the cytoskeleton. So within the individual neuron, the cytoskeleton is very important for its function for total brain function. So here I have the red arrow for the axon, think of information going out, and the blue arrows for the dendrites, for this hippocampal neuron, for the information going in. And to have that total brain function, all these axons, all these dendrites, all these extensions have to find their proper partner, have to navigate to where they need to go. Now, within the brain, there are multiple ways in which this happens. What I focus on specifically is semaphore and 3A. So semaphore and 3A is an interesting secreted actual chemical cue because it has two different effects on axons and dendrites. Whereas in axons, it functions as a negative growth cue, causes the collapse of that growth cue via the oxidation of the actin within that growth cone. And then within dendrites, it has the opposite effect, where it functions as sort of a mild positive growth cue. And part of that actual effect requires the reduced state of soluble guanylate cyclase as well. So we kind of have this question of, okay, are the axons, do they just typically maintain a more oxidized state than the dendrites in the hippocampus? And using a redox-sensitive fluorescent dye, I was able to look at 3 different time points of hippocampal neurons. And surprisingly enough, if it turns out that there actually isn't a significant difference. So you can say that there are some cells, the axons are more oxidized, more reduced, but overall there isn't a difference. The overall kind of balances out kind of that ratio of 1, being that the axons and dendrites themselves are the same. So then I asked the question of, okay, well, there are some cells where the axons are more oxidized and more reduced. Do I see a difference in as far as the actual morphology? Are some actually just more mature or more developed? So in order to do that, start off the Scholl analysis. So again, I have a cell here, multiple diameters all around it. Then I just measure how many times neurite actually crosses one of these diameters, and find this actually the same thing. So just for a bare Scholl analysis, there's no difference between those with the most oxidized or most reduced axons. Same thing for the branching index as well, which is a kind of another measure of that Scholl index. And so far, there actually doesn't seem to be a difference, which is surprising based upon our initial hypothesis. And thank you very much. Any questions?
Thank you, Miles. Are there any questions?
Hi, Miles. That's very nice talk. I have a question whether you have tried to use another method to analyze oxidative level. For example, isolating the dendrites and the axons and then subject them to proteomics, metabolomic analysis.
So that kind of actually goes into what I will be continuing to do, where I will be separating the both axons and the dendrites, and then actually exposing them, though that alone and not the somas, to different redox states as well, so I can manipulate them just by themselves as well. So I'm not doing proteomics, but yeah, that general idea of kind of isolating the dendrites and axons and seeing how that's affected.
Great, thank you. Are we ready to move on? I believe Taylor is next.
Yep. Hi, my name is Taylor Jorgensen, and I'm also a member of Martha Gillette's lab, and I'm co-advised by Dan Yano and Justin Rhodes. And my project involves astrocytic gap junction dynamics in the rat hippocampus. So, as you probably know, the hippocampus is a major site of learning and memory consolidation in the brain. But we also know that the hippocampus shows circadian patterns that are coupled to the suprachiasmatic nucleus, or SCN, that is known to be the master clock of the body. So there are many processes of the hippocampus that show this connectivity, but just a few of them here. We see that membrane excitability and neuronal activity changes between day and night. We see that redox state oscillates in the hippocampus in approximately 24-hour cycles, and also that this oscillation persists in brain slices. And then we also know that hippocampal long-term potentiation, or LTP, shows a greater enhancement and slower decay at night than when compared to the day. And this is one of the major experimental models of learning and memory. So specifically, the type of cell that I'm interested in is the astrocyte, which is a type of a bundle glial cell that participates actively in the tripartite synapse. They're known to have many diverse functions, but they modulate neuronal activity and also synaptic plasticity. But astrocytes don't work on their own. They actually form networks of astrocytes via gap junction coupling. And so this happens when two connexin hemichannels on adjacent cells couple together. And this allows for molecules of less than 1 kilodalton to pass through the cells. And these networks of cells become metabolically and electrically coupled. Specifically in the dentate gyrus of the hippocampus, our lab has found that astrocytes show a diurnal coupling pattern. So the way this was found was by using a patch clamp technique where astrocytes were using their voltage and their currents, they were identified and then injected with the sulfurotamine B dye, which can permeate gap junctions because of its size. So by injecting this dye into one cell and counting the number of cells in the network, we found that in the evening, there are more cells in network compared to the morning. So now that we know about the differences between night and day, the next thing that I personally want to look at is how inhibiting these gap junctions at different times of day affects the networks. Will we see the same magnitude of disruption during the day and night, or will we see different percentages of cells becoming uncoupled? And after we further characterize these networks and their inhibition, I want to then quantify the changes between morning and night and see how gap junction inhibition affects the neuronal activity. Thank you.
Great, thank you, Taylor. Are there any questions?
I have a quick question. I assume that when you're making these slices, you're separating the SCN from the hippocampus. So if you look at astrocytes, do they have expression of clock genes like BMAL or PERT?
I know that the neurons in the hippocampus specifically do show these clock genes. I am not 100% certain if we see these clock genes in the astrocytes as well.
Thanks.
This is about it. Oh, sorry, go ahead. I was got a quick question about, can you reset these circadian rhythms with other light dark cycles, or does that not work?
So there are drugs that we can use to synchronize cells to make them all the same. And also we have to be really careful when we are taking the slices and the brains out, because when you cut the optic nerve, that can also reset. So these cells can be reset and changed on their cycles.
"30 seconds left for other questions."
Oh, so is this one also happen in human, maybe in a opposite direction? So maybe in the maybe early day rather than early night, for example, gap junction coupling in the TG?
Are you asking if this also occurs in humans?
Right.
I have not looked into any of the human studies, if there are any, but it is worth noting that nighttime in rats is actually their active cycle because they are nocturnal.
Right.
that's-
That's what I was trying to ask, whether, yeah. Okay.
Great. Thank you, Taylor. Will, you're next.
Okay. Hey, so hello everyone and welcome to my presentation entitled Engineering of Mesenchymal Stem Cells for an In Vitro Blood-Brain Barrier Model. My name is Will Baker and I'm advised by Drs. Kong and Leckman. So the overarching thrust of this research project is the enhancement of the secretion activities from mesenchymal stem cells, or MSCs, in order to promote the formation of a blood-brain barrier microfluidic model, an in vitro model for the study of drug transport. Now, in this chip, we seek to partially recapitulate the blood-brain barrier by facilitating a co-culture of blood-brain barrier constituents, namely endothelial cells, astrocytes, and parasites. And in addition, we also see neurons, as well as our MSCs, all in a 5-channel microfluidic chip. Now, importantly, we see our MSCs in the channel adjacent to endothelial cells because we utilize these MSCs to promote the formation of endothelial cell vasculature. So you may ask, okay, why are we using these MSCs? Well, we have identified these cells because they show prolific secretion of cytokines that promote neovascularization, in particular angiogenic growth factors such as vascular endothelial growth factor. Now, unfortunately, one major limitation is that MSCs are associated with attenuated paracrine secretion in vitro. Therefore, we seek to devise ways in order to improve the secretion activities of MSCs to promote endothelial cell vascularization. And specifically, we seek to do this via cytokine preconditioning of our MSCs. To that end, the Kong lab previously showed that activation of MSCs with the cytokine tumor necrosis factor alpha successfully improved the provascularization effects of MSCs, specifically via upregulation of angiogenic factor production. This provascularization effect was verified in a microfluidic angiogenesis assay similar to the design we proposed for the blood-brain barrier model, with the major exception that this microfluidic chip comprises a co-culture of MSCs and endothelial cells. Now, unfortunately, one issue is that tumor necrosis factor alpha can be associated with a pro-inflammatory effect. So we decided to try and identify an alternative ligand that could provide a more immune neutral effect on MSC secretion activities. And accordingly, based on the literature, we identified specifically fibroblast growth factor 2, or FGF2, as this ligand is commonly used in various cell medias as
30 seconds.
proliferative growth factor. And in fact, we demonstrated that preconditioning of our MSCs with this FGF2 induced a similar increase in angiogenic growth factor secretion to our tumor necrosis factor alpha. And this was evidenced in increases in vascular endothelial growth factor production. Now, going forward, our next step is going to be to utilize a similar angiogenesis on a chip design as we did here, this time with our FGF2 stimulated MSCs, in order to determine whether or not this ligand is actually capable of inducing MSC secretion to an extent that actually permits endothelial cell vascularization, and therefore would render this ligand suitable for our blood-brain barrier model. And with that, I thank you, and I will take any questions.
That was interesting. Can I ask a quick question? It's
Yeah.
maybe about the previous studies, maybe more so than your current studies, but was there some optimal level of TNF alpha that would prime it? Because if you gave them too much, would it be too bad for them? Or was the delivery method able to modulate that there was never going to be too much that was delivered that would be toxic?
Yeah, so specifically in this instance, it was actually a liposome nanostimulator delivery vehicle that released tumor necrosis factor alpha in a sustained manner to prevent excessive dosages of tumor necrosis factor alpha.
Any other questions? Thank you. Charles, are you ready to go?
So I'm Charles. I'm in the Sutton Lab. My project is on MRI perfusion imaging. So the method we use to measure perfusion is arterial spin labeling, or ASL. This quantifies the perfusion in physical units, so we can get the volume of blood that enters a given mass of tissue within a specific amount of time. This method requires no exogenous contrast agents. So in the figure at the bottom, on the left is a typical MRI water density weighted image. So we first suppress that signal using radio frequency pulses. And then we label the blood upstream using more radio frequency pulses. So there's no exogenous contrast needed. And you're labeling the spins in the arteries here, which is why it's called arterial spin labeling. And then that blood in the arteries flows up into the brain. And that's where we can get our contrast from. And a major limitation with this method is that it has a very low SNR, signal to noise ratio, resulting in large voxel sizes. And we want to use a method called partial separability to improve that. So partial separability refers to the fact that the temporal data is partially separable from the spatial data. And this implies that the data is compressible. So we do not need to fully sample all the data for all data points, because every data time point is a MRI perfusion image, and these time points are correlated. So we don't need to collect 100% of that data. And what doesn't have a spatial temporal correlation is random static, which is noise. So this also acts as a denoising procedure. And we applied the PS reconstruction to this ASL MRI for the fully sampled data acquisition, which is on the bottom left. And on the right is what we hope to achieve, partial separability sampling, which is needs much less sampling and go faster. So here are results for, this is fully sampled data. This is a standard reconstruction on the left, and in the middle is the partial separability reconstruction. And you can see from looking outside the brain that the spatial SNR is higher in the PS reconstruction. And on the right is the temporal SNR of the PS minus the temporal SNR of the standard. You can see based on where 0 is on the grayscale color bar that we have a higher SNR using the PS model. So next we hope to achieve the data acquisition using the sparsely sampled data. And this should make the images sharper as well as shorten the scan time. And then we hope to apply this to blood cerebrospinal fluid barrier imaging so we can measure how much CSF is being created from the blood.
Great, thank you, Charles. Are there any questions? All right, let's move on to Olawale.
Hello, my name is Wally Salahuddin. I'm a third-year PhD student in computer science, and today I'll be presenting my work with Aaron Barbe, Brad Sutton, and Sami Koichal on nuisance artifact removal in fMRI. So just as a summary, fMRI is used to examine neuroactivity in the brain by measuring small changes in magnetic field induced by blood flow that occur after neurons fire. However, due to the spatial temporal resolution of fMRI, it's highly susceptible to nuisance artifacts, such as those from respiratory, cardiac, and motion-induced changes in the magnetic field. So it turns out that given a 4D fMRI scan, we can use a method called independent component analysis, or ICA, to decompose the spatial temporal data into K-independent spatial maps of the brain with unique associated time signatures. And so what we know is that these independent components can often isolate nuisance artifacts with known spatial temporal signatures. For instance, the effect of motion tend to be most pronounced around the edges of the brain. These signatures also occur in the temporal domain as well. So previous work by Krim et al. Gives an automatic model to examine these spatial maps and time signatures to identify components that are consistent with known signatures of nuisance artifacts. And we're able to remove the effect of these independent components from the original scan in hopes of honing in on the neural signals of interest. So the natural question is, what about the signatures that are unknown? What can we do about them? And this is where our work comes in. So we propose that the known nuisance signatures give us an insight into more complex ones. Specifically, we expect unknown nuisance artifacts to share some probabilistic structure with the known nuisance artifacts. And we aim to identify this structure in a data-driven fashion. So given that we have k-independent components and some known nuisance components from previous work, we develop a probabilistic graphical model that encodes the structure of how the k-independent components relate to each other. And then once we find this structure, we identify independent components that may not exhibit the typical nuisance signature, but are in the neighborhood comprised of identified nuisance components. And then we're able to relabel these independent components as nuisance, and then also remove them as well from the original scan, hoping to hone in on the neural signals of interest. So our preliminary result examines the efficacy of our method on predicting general intelligence in the U.S. Army men from resting state connectivity in 7 known networks in the brain.
30 seconds.
So relative to the original scan, we can see that the current state of the art, if you will, IC aroma gives us a 1.3% reduction in root mean squared error in predictive error, and our group-specific graph signature gives an 8.7% reduction in error, and our individual-specific graph signatures give 3.8% reduction in error. So I'll note that as we go down this table, the methods remove increasing amounts of information from the fMRI scan. So the fact that we were able to see a decrease in predictive error indicates that our method does indeed remove nuisance artifacts that hinder inference about general intelligence. So our next steps are to better understand the results of this data set, including some of the degrees of freedom left and the statistical power left in the signals after our methods is applied. And we would also like to evaluate the cross-group efficacy of this group-specific graph signatures that have emerged, specifically looking at how they extend to the Human Connectome Young Adults Project data set. And we would also like to generalize this algorithm beyond ICA decomposition, and this would include, for instance, PCA. And that's all I have. Thank you, and I'll take any questions.
Great, thank you, Wally. Are there any questions? All right, let's move on to Jenny.
Okay, sorry, I was muted. Hi, my name is Jenny, and today I'm going to be talking about exercise and the brain, and more specifically, how hippocampal activation plays a role in that exercise-brain relationship. Oops. So there are 2 main hypotheses that aim to identify how exercise exerts its beneficial effects on the brain: the muscle hypothesis and the brain hypothesis. And there's a vast amount of literature covering the effects of various muscle factors that are released into the blood, cross the blood-brain barrier, and exert an influence on the brain as a result of that exercise. So today I want to talk primarily about the brain hypothesis, which states that a particular brain region called the hippocampus is activated immediately prior to and throughout the duration of exercise, and is also activated proportionately to the intensity of exercise. This activation of the brain itself is hypothesized to contribute to the beneficial effects of exercise on brain structure and function. So in order to test this hypothesis, we designed an experiment in which we had 2 mice, a treatment mouse implanted with a red micro-LED into its hippocampus, and a control mouse implanted with an inactive mock micro-LED into its hippocampus. These were transgenic mice with red-shifted channel rhodopsin, so their neurons would respond to red light. These mice were allowed to run freely on running wheels for several days while being fed a tamoxifen diet, which would allow us to permanently label the neurons that were acutely activated during wheel running. Then the mice were removed from the running wheels, injected with BRDU, which allowed us to measure neurogenesis, and we optogenetically stimulated the hippocampal neurons that had been previously activated during running in a pattern intended to mimic the neural activation that occurs during normal wheel running, but this time in absence of any actual wheel running. So to measure activation of the hippocampus, we analyzed the numbers of CFOS positive cells in the hemisphere where the micro-LED was implanted compared to the opposite hemisphere, and found that for the mouse implanted with the red micro-LED, there was a significant increase in activation in the portions of the hippocampus that were closest to the micro-LED compared to the portions that were farther away. However, for the control mouse with the mock micro-LED, there was no difference. So this demonstrates that we were able to successfully activate the hippocampus using the red light.
30 seconds.
In terms of neurogenesis, we found that both mice actually had less neurogenesis in the regions of the hippocampus closest to the implanted micro-LED, and the difference between hemispheres was actually greater for the treatment mouse implanted with the red micro-LED. So our conclusion from this is that the decreased neurogenesis is likely due to damage to the tissue caused by the implantation of the micro-LED. And thank you to my advisors and our funding sources, and of course, MBM.
Great, thank you, Jenny. Are there any questions?
Hey, Jenny, very nice nice talk. So did you check by histology if there is really increased cell death or the volume of the hippocampus? Is there any change in that aspect?
Yeah, we didn't actually measure that in this case, so I don't know. Yeah.
I'm very interested in this project, so maybe if you want to, we can talk a little more later.
Oh yeah, definitely.
Yeah.
I would also add that maybe you might want to consider using the step function options that can be activated through the skull. So you don't have to put the LED in, you can activate them through the skull. So it might just be a way that you can do it without inducing any damage.
Would you be able to do that for just the hippocampus, or would it be for more cortex?
No, but if you target your virus to just the hippocampus, then it probably would be fine.
Oh, okay. That's really interesting. Thank you.
Are there any other questions? All right, let's move on to Shannon.
All right, does this look good? Okay.
Yeah.
Hello, I am Shannon Bernash. I'm a student in Jonathan Sweetely's lab, and my co-advisor for the MBM program is Martha Gillette. And I'm going to talk a little bit about my research, which focuses on the glymphatic system. The glymphatic system is a system that is believed to clear fluid and soluble molecules from the brain. This may include waste molecules. So this makes it very important and potentially harmful if there's any disruption of the system. So it has potential implications in disease, such as Alzheimer's disease. And interestingly, the system is most active during sleep. So this makes it important to consider circadian rhythm when studying the system. These are the questions I'm hoping to answer with this project. We are hoping to find out which peptides are cleared from the brain by this system, and also how circadian rhythm disruption affects this system. In order to answer these questions, these are the methods we have been using. First, we are collecting cerebrospinal fluid from rats. Actually, my collaborators in Martha Gillette's lab are doing this part. We are looking at two different variables, location and time of day. So for location, we are collecting fluid both before and after it passes through the brain. Before it goes through the brain, it is first produced at the choroid plexus. So we are sampling from near there, and then the fluid actually passes through the brain, clearing waste molecules, and we are sampling from either a lymphatic vessel or sinus, where we can then compare the composition of the molecules in the fluid
30
and
seconds.
see which ones were cleared. And we are also looking at time of day. So we want to know how circadian rhythm disruption may affect this system's ability to clear molecules from the brain. And in order to analyze these samples and identify the molecules in them, I'm using a technique called capillary electrophoresis coupled to mass spectrometry. This is an analytical technique that separates the molecules in the samples so that we can identify them, and it allows us to identify them by giving the mass to charge ratio of the ions produced from the samples. And I'm specifically looking at the peptides contained in these samples. So I'm using a peptidomics approach. I will be searching the mass spectra I acquire against a database from the species Radis norvegicus to identify the peptides contained in these samples. And hopefully we'll be able to find out how the lymphatic system's function is affected by the time of day as well. I'd like to thank these people as well. Are there any questions?
Great, thank you, Shannon. Are there any questions? All right, let's move on to Samuel.
Okay. Hello, everyone. I am from Jonathan Swedler's lab, and my co-PI is Professor Martha Gillette, and I'm going to present on the amino acid containing peptide discovery in mammals. Now, neuropeptides are cell-to-cell signal molecules that are actually obtained from prohormones, protein prohormones, and the activity of these neuropeptides have been actually implicated in several organ functioning. Some of these neuropeptides, in order to get the active forms, you have to go through a series of post-translational modifications. One of the post-translational modifications that has been recognized is the conversion of one or more of these L-amino acids to a D-amino acid. And the most advanced technique that is being employed recently in peptidomic studies is mass spectrometry. And as you can see within this peptide here, where the only difference is the changes in the chiral center right here, that is from an L to a D form. There is no mass difference, so therefore mass spectrometry cannot actually be able to easily distinguish between these peptides. However, mass spectrometry still remains one of the state-of-art instruments that you want to employ because of its high accuracy. So in our lab, we've employed mass spectrometry together with reverse phase chromatography and some chiral separation columns, together with ion mobility profiling and also some protease enzymatic assay to identify the amino acid containing peptide in several other organisms such as mollusks and planarians. Now, my work here is to try to employ some of these techniques and apply some modifications where possible to actually discover some of these amino acid containing peptides in mammals. By employing these techniques to an extract from the hypothalamus of rats, we actually observed that galanin, which is a peptide that is implicated in feeding, sleep, and wakefulness and other activities, have a split eluting peak, as you can see from the extracted ion chromatogram right here, that it has two peaks at slightly different retention times. And when we try to look at the tandem MS for these two peaks, they have the same tandem MS, meaning that they come from the same precursor ion. Now, the interesting thing here is that although these peaks are the same, coming from the same precursor ion and the same peptide, they have different ion mobility profiles at different charge states. So this slightly different ion mobility profiles tells us that they may have different gas phase conformation, which also give us an inference that this galanin may be occurring in different isomeric forms.
30 seconds.
Now, in order to ascertain that whatever we are observing is not from our extraction procedures, we try to employ synthetic standards and treat our samples with a synthetic standard with extraction conditions. And we observe that there is no difference between the treated synthetic standard and the untreated one. So therefore, whatever we are seeing is something that may be occurring endogenously within the tissues. So in summary, we have actually observed that guanine may occur in different isomeric forms, so it becomes necessary to quantify the levels of these isomers if indeed they are present. And if they are present, what are the functional roles of these different isomers? It becomes interesting to ask some of the questions of the functions of guanine. And also we try to extend these techniques to actually discover several other peptide stereoisomers in mammals. Thank you.
Great, thank you, Samuel. Are there any questions?
So are there any examples of the different function of this L or T form amino acid containing the neuropeptide?
Yeah, there are several functions that have been attributed to them. With some of them, the D form is active and the L form is inactive. With some of them, both of them is active. So it depends on the peptide that you're actually looking at. And some of them too, one of the things that has been implicated is the stability. Once the conversion happens, they mostly becomes more stable to protease activity, so they can last longer. And that is one thing that we are looking at. So the stability of the peptide, and then also some of the activities, the D and the L form may have the same or slightly difference in activities.
Any enzyme isomerase, you could use those knockout animals to really use as a kind of control experiment.
Yeah, there is a person in our lab, Harvey, who is actually working on the isomerase activity. It's kind of challenging because there is a whole lot of things that need to be done to get the isomerase itself and how it actually works. So even isolating the isomerase has become challenging, but there is a couple of people working on the isomerase activity to determine the actual isomerase that is performing this function. Some of them has been discovered in frogs. And I think there is 2 isomerase or 3 of them that has been reported, but there's this confusion in literature that some of the isomerase reporting may not be actually catalyzing the function that they are assigned to. So identifying the isomerase itself has become a little bit challenging, but we're still working on trying to identify the isomerase, then it becomes easy for us to do the knockouts to identify their functions.
Great, thank you, Samuel. Let's move on to Jenny.
Sorry. All right. Hi, everyone. Today I'm going to be talking to you about my research looking at the pharmacological inhibition of STEP protein and its effect on kainate-induced seizures and hippocampal excitability. So foremost, anti-epileptic drugs or AEDs do not work on everyone, especially patients with temporal lobe epilepsy or TLE, and they account for about 60% of all epileptic patients, and their seizures are typically drug resistant. So finding small novel chemical compounds are in critical need. And my particular compound of focus is TC2153, which is a novel STEP protein inhibitor. STEP is encoded by the PTPN5 gene and is expressed widely throughout the CNS, particularly in the cortex and hippocampus, which is where seizures in patients with TLE arise. And varying levels of STEP expression are associated with many types of neurological disorders, not limited to Alzheimer's, epileptogenesis, and schizophrenia. So I personally was looking at TC compound's effect on seizure susceptibility, and I looked at a kainate-induced seizure model in wild-type mice, where I injected these mice with TC compound and 3 hours later injected them with a chemoconvulsant called kainic acid to give rise to seizures from the hippocampus. And looking at both male and female cohorts of mice over a 2-hour time period, we see that there's a decrease in seizure susceptibility in TC compound treated mice at several time points in males, but this is significantly decreased across the board over the 2-hour time period in females. And this led us to wonder whether or not there was a sex-dependent difference. when using this compound. So with our collaborator, Dr. Catherine Christian, we ovariectomized a female cohort of mice to abolish circulating female sex hormone, performed the same injection procedure, and found that there was a rescue in this decrease in seizure susceptibility after giving the TC compound. We also specifically wanted to look at the effect of TC compound on hippocampal excitability. So we took hippocampal brain slices from GCAMP6S mice, and these mice express a genetically encoded fluorescent calcium indicator, where when there's a positive influx of ions into the cell, this excites the cell and causes a conformation change that pushes up a fluorophore, and it fluoresces, and we can measure that fluorescence intensity. So after incubating in a potassium chloride bath, slices that were either incubated with vehicle control or TC compound.
30 seconds.
Thank you. We can see that there's a significant decrease in this fluorescence intensity in the TC compound treated slice. And we see this in several different regions of the hippocampus, including the dentate CA1 and CA3 regions. All right, and our future goals are to look at whether TC compound has similar effects in human models, such as human iPSC-derived neurons or human brain organoids, to see if it's a translatable drug to use in humans with epilepsy. And with that, I would just like to thank my lab and my collaborators, and I'll take any questions. Thank you so much.
Thank you, Jenny. Are there any questions? All right, let's move on to Ryan.
Can everyone see this okay?
Yes.
Okay, sounds good. So my name is Ryan Miller. Today I'll be talking about ROS responsive antioxidant regulation of senescence propagation in mesenchymal stem cell-based therapies for neurodegeneration. So as many of you know, neurodegenerative diseases are a broad class of disorders characterized by progressive neuronal death. One approach to the treatment of these diseases is through mesenchymal stem cell therapy. MSCs have been shown to secrete neurotropic growth factors and has also been shown to modulate the immune system and local inflammation. While MSCs present many beneficial properties and have been included in over 900 clinical trials as of 2018, in vitro culture and therapeutic efficacy can be hindered by senescence propagation driven by reactive oxygen species. When in a senescent state, MSCs exhibit decreased exosome production and growth factor secretion, which is essential for therapeutic efficacy. As such, the focus of my work has been on designing ROS responsive antioxidant delivery systems that can maintain proper ROS homeostasis. My system comprises of two parts. N-acetylcysteine is the active pharmaceutical ingredient. It can scavenge reactive oxygen species, and an ROS responsive stabilizing polymer that can promote both crystal aggregation and crystal anchoring to MSCs. So it's not going forward. There we go. Oops. So in order to obtain a narrow particle size distribution that is necessary for consistent release properties, I turn to drop microfluidics that can produce micro-sized single emulsion drops that function as individual reactors for my crystallization processes in the video. Displayed in the histograms, crystallization in the droplets leads to drastically narrowed particle size distributions, and the addition of thermocycline to control nucleation events also improves the size distribution. The activity of the NAC crystals is also important. First, I confirm the release of NAC can be modulated by the presence of reactive oxygen species. Plot looks at NAC dissolution as a function of time and illustrates that following hydrogen peroxide exposure, the NAC release increases, notably the light blue and the gray curves. I also wanted to confirm that ROS scavenging potential of these crystals in vitro. The confocal images depict the intracellular ROS levels using a dye that fluoresces green when oxidized and illustrates the improved efficacy of the crystals compared to NAC solution in no treatment conditions after cells were placed into an oxidative environment. So from here, My current work is focused on more in vitro characterization, but this time of senescence propagation in MSC culture and the protective effects of NAC crystals. So that concludes my talk, and I want to thank both my advisors along with the MBM program.
Great, thank you, Ryan. Are there any questions?
So Ryan, I have heard you give this talk before, but it never occurred to me before that this might be something that could be applied to Miles's work. Miles, are you there?
For sure.
I know, Ryan, you've heard Miles talk before.
Yeah, I didn't show all my results because of the time limit, but these crystals actually have pretty good control when I control the ROS responsive linker onto the rate that they dissolve in the presence of ROS, as well as what range of ROS they become active. So if we're using like 0.1 micron levels, I would have to increase the amount of, or decrease the amount of selenocysteamine, or vice versa if I'm using higher concentration. So it's definitely tunable. And if Miles thinks he can use it for his work, I can produce these at a pretty large scale.
Well, Miles didn't say this. I hate if Miles is there. I'd rather he said it, but nevertheless, he's developed a microfluidic device technology that lets him isolate different subunits of the cell. So that sounds like, a great idea to me anyway.
Sounds good.
Yeah.
I'll tell Miles to get in touch with you. How about that?
Okay.
Yeah, sorry, I was double muted.
Oh, there
Yeah,
you
that's
double
fine.
muted. Oh dear. Okay, I'll step back now, Miles, and you can speak.
But yeah, we'll have to get in touch with that then.
Yeah, definitely. Any other questions?
All right, let's move on to Sarah.
All right. Let's see. Pointer really quickly. Okay, so good morning, everybody. I'm Sarah Bell. I am co-advised by Jonathan Sweedler and Rashid Bashir. And today I'll be focusing on one of my projects, specifically about high throughput single cell multi-mass spectrometry. So this is a collaboration with Allison Bell's lab on campus, where we're interested in understanding the parenting behaviors of these stickleback fish. So these fish are interesting because the males are the main caregivers, and their parenting behaviors have actually been shown to differ both within populations, so like from individual to individual, and across populations, with one of the more stark differences being between the white fish, which is a very unattentive father, and the common fish, which is a very attentive father. And so, as you may expect, these behaviors do have an effect on the offspring, both on their fitness and their own parenting behaviors whenever they have their own offspring. And so the goal of this project is to link transcriptomics data from the Bell lab to whatever peptides are actually playing an active role in maybe regulating these behaviors. Now, the way we plan to look at these peptides is using high throughput single cell mass spectrometry. Now, how we do this in the Sweedler lab is we isolate our tissue of interest. Usually we use the brain. Currently, we're using the pituitary for these fish, and we enzymatically dissociate thousands of cells, stain them and plate them for fluorescence microscopy. And then from that microscope image, we can perform a task using a program called MicroMS from our lab, which actually allows us to pinpoint the exact pixel location of these individual single cells within the image, and then translate that to a physical location within the mass spectrometer. This way we can directly target our single cells with our multi-laser to produce mass spectra. Now, I've done this for 40,000-ish cells from 20 fish so far. And the reason we're doing it on such a large scale is so that we can categorize these cellular populations based on the variability of their peptide profiles. Now, we know that their peptide profiles will vary just based on the heterogeneity of cellular populations within tissues. So we can capitalize on that heterogeneity and use a machine learning classification task recently produced in our lab to determine which of these peptides that we're seeing in MALDI-MS may be most indicative of the different behavioral types. So we hope to produce a peptide list for the white fish and another peptide list for the common fish, and then take those peptide lists, compare them back to the behaviors and the transcriptomics data from the Bell Lab. In this way, we hope to better understand these behaviors. And with that, I'll take any questions.
Great, thank you, Sarah. Are there any questions?
I have a question. If no one else does, I can always come up with a question. What is the resolution of peptides? I mean, you must have some sense, for instance, in a certain tissue of the fish, what peptides are expressed. And the single cell proteomics, how comprehensive do you think the assessment is?
So for proteomics in general, like obviously we use a LCMS-based method, which actually produces a peptide library, because for both of these, we're using different ionization sources. And of course, we're going to have missing values. So one method is going to miss some peptides and the other method will get those. So we do use kind of a complementary two-prong approach in that way. But the reason why we do it on the single cell level is not because we're going to comprehensively get all the peptides that way. It's because we'll see more variability, and hopefully we'll be able to see some peptides that are lower in abundance and would be drowned out in bulk analyses. So if that answers what you were going at.
It does. Thank you.
Yeah.